Um, what I'd like to do today, a couple of things. First of all, you've had a worksheet. It's a short, fairly simple worksheet for a while, and by now you've all studied it, looked at the questions, and if you remember last time, I suggested that instead of just reviewing the worksheet, I call on individual people randomly, arbitrarily, and ask you to go one by one through the questions. So I want to try that today, see how it works. And if I don't call on you today, because there aren't enough questions to go around, I'll try to get to you sometime in the future. Okay, so first of all, I want to get the worksheet up. You should all see worksheet, do you? In letters large enough that you can read it?
Yes.
Okay, so we're going to start with question number 1. I'm going to start by calling it a name. Let me call James Campbell. So James, could you read the question and then read the choices and tell us what you think?
Which of the following is true about enzymes? A, they speed up chemical reactions. B, not destroyed by the chemical reaction. C, lower activation energy. Or D, all of the above. The answer is D, all of the above.
Everybody agree with that? Anybody disagree?
I agree.
I agree.
Okay, so do I. Very good. So let's go to number 2. And is Sadie Leash here? Sadie's not here. Okay, how about Melissa Bates? Is Melissa here? Michael Korn?
I'm here.
Okay. Do number 2.
All right. Which of the following is not true about enzymes? Accelerate chemical reaction rate. B, enzyme shape is important for function. C, inactivated by reaction that is mediated. D, reduce energy required for reaction to occur. I need a second to think.
Take your time.
It's C, inactivated by reaction that is mediated, I think.
Okay. Anybody else think that? Anybody object? Well, I agree. It is. It's not true that an enzyme, and that's a part of the definition of an enzyme is that it's not changed by the reaction. So if it's active before the reaction, it's equally active after. Okay.
So which letter was that?
Number C.
Let's see. Could I answer number 3, Professor?
Yeah, what's your name?
Veronica Munoz.
Veronica, okay, go ahead.
An enzyme is a blank: A, carbohydrate, B, protein, C, lipid, or D, nucleic acid, and it is B, a protein.
Yes, it's protein. So, yeah, all of the major macromolecules are listed here, and an enzyme is just one of them. And none of the others have anything whatsoever to do with catalysis. Okay, which of the, well, I won't read it. I'll pick a name first. Let's see. about Nathan Huzar?
It's number 4.
Number 4.
Which of the following affects enzyme activity? A, temperature, B, ionic conditions, C, pH, D, all of the above. I think it's D.
Yeah, it is. It is D. Okay, so let's go to number 5. Who's the lucky person that gets number 5? Is Santazaya here? Georgiana Willis? Okay, Erica Arara.
I'm here.
Okay,
Um,
you do number 5.
So the graph shows the progress of an enzyme reaction. Which letter on the graph would best be labeled activation energy with enzyme? I think it would be C. Letter C.
Do the rest of you agree? Well, you are right, Erica. The B would be the activation energy without enzyme, and A and D are just the beginning and the ends of the curve, the reaction curve. Okay. On number 6. Teresa Medina, number 6. Teresa, do you have, can you hear me? Apparently not. Amir, can you do number 6? People must not be hearing me. I don't think I'm muted.
I can hear.
Is that Amir?
No, but I'm just like
I can
Oh,
do number 6.
you can hear. Yeah, okay. Do I have a volunteer who hasn't spoken yet to do number 6?
Yeah, I can do it.
And you are
Jordan Farrell.
Jordan Farrell. All right.
Cellular structures responsible for converting carbohydrates into usable energy are mitochondria.
Yes, that's right. Now, this is a little bit premature, and the rest of the questions on this worksheet, I got ahead of myself. They haven't really been covered yet, but we're going to go through them anyway because they will be. And so, yes, the answer to number 6 is mitochondria, and we'll talk a lot more about that in today's lecture and the one beyond that. Okay, do I have a volunteer for number 7?
I can do number 7.
That's Violet.
Melissa Bates.
Oh, Melissa Bates, okay.
Most
right.
of the energy from cellular respiration is converted into, and it should be CATP.
Yeah, that's right. And again, we really haven't talked about cellular respiration yet, but we'll get into the mechanism of how that is done. It's not sugars. Sugars is what is the substrate for making ATP in respiration. Now the next one, we haven't covered at all, but we will. But would anyone volunteer to do number 8?
I can do number 8.
And your name?
Melody Garcia.
Melody Garcia, okay.
Okay, so it says, under which conditions would a cell begin lactic acid fermentation? And I think the answer is A.
Answer is A.
Yes.
Does anybody else have an opinion about the answer number
I
A?
think it's C, anaerobic.
Anyone else? Okay, the answer is, in fact, C. Under anaerobic conditions, the cell will start to make lactic acid instead of its normal output. Okay, number 9 is related to number 7. Fabian, can you do that? Is Fabian there? He was here. Maybe he's gone. Okay. Somebody volunteer, please. Isabella Estrada.
Yeah, it says breakdown of blank is done by mitochondria to provide cellular energy, and I think the answer is D, glucose.
Yes, you're right. The answer is glucose. So very good. You've done well on the questions that you haven't really had the background for. So that's very, very well. This was an experiment trying to do a worksheet in this manner. What do you think? Anybody like it or anybody doesn't like it?
I thought it was very useful.
Okay, well, I like it that I'd like the class to be more interactive, and this is one way that we can try to do that. Yeah, I like it better than me just sitting here reading them off myself. Okay, anything anybody has to say about any of these questions? I will try to get another worksheet out that will be more comprehensive sometime this week. Don't forget that on Monday, we have quiz 8, and that will be focused primarily on enzymes and energy. Most of the questions are on that. I think there's a little bit on photosynthesis, but it's pretty fundamental. So if you know a little bit about photosynthesis, which is what we're going to talk about today, and a lot about enzymes and energy, you'll do fine on quiz 8. That's Monday, and again, you'll have all day, 15-minute time, and you'll have it until midnight on Monday evening to do quiz 8. All right, I'm going to
I sent you a chat message, sir.
Okay. I can't find my chat here. What does the message say?
It was, I don't know, maybe a fine point about question 9.
What is it?
It says that glucose is processed. The breakdown of glucose occurs in the mitochondria, but doesn't the breakdown of glucose, the glycolysis, actually happen in the cytoplasm, whereas the pyruvate product is what's then sent into the mitochondrial matrix to be processed?
So say that again. You're saying glucose oxidation starts in the cytoplasm? And
Glycolysis, yes, the breakdown of glucose into pyruvate occurs in the cytoplasm, but then that pyruvate goes into the mitochondrial matrix into the citric acid cycle.
Okay.
I
Yeah,
was just saying that seems a little misleading to me.
perhaps it is. I'm not sure about this, but we'll check on it. You are right that pyruvate is the immediate product of glucose, and that's where the energy comes from. But where that starts, I'm not sure. Okay, good point. Thank you.
Like I said, it's just a fine point, and that's why I sent it in a chat. I'm sorry you couldn't find your chat. I didn't mean to distract the whole class with it.
Okay.
I can ask a question, sir.
Yeah.
I'm sorry. I had the trouble hearing you the first time. What these sheet? You tried to explain to us. I'm sorry.
Say your question again.
My question, the sheet you already, shown to us for what this sheet?
Did you not receive this worksheet?
No, I don't receive nothing. And then I had a trouble the first time when I got in the class to hearing anything.
Oh, okay. So that's why you didn't respond when I called your name. I sent this worksheet out as an announcement to the entire class, so I don't know why you didn't get it. I will send, I can send it to you again. Make a note to do that. And there should be another worksheet coming within the next day or two. So make sure you get that. If you don't get one, send me a note and let me know.
All right.
Over which subject?
The subject of the next worksheet will be photosynthesis and respiration. Maybe the emphasis will be on photosynthesis because that's what we're talking about today, and maybe a little bit on respiration because also that will be included in today's discussion. You should have the overview slide showing. Tell me if you don't.
Yeah, we can see the slideshow.
You can see it, okay. All right, again, this is just to remind you of the context here and how we're trying to tie these 4 different topics together and make them all make sense. And it all starts with energy. Energy, where does it come from? And how do life forms use energy? Enzymes, because enzymes are essential for all the metabolism, all the chemical reactions that go on in a living cell. Photosynthesis, which is our sole source of energy, the capture of energy from sunlight. And photosynthesis miraculously converts that captured sunlight into food. And finally, respiration is the derivation of energy from food. And so the emphasis up till now has been on energy and enzymes, and now we're going to do photosynthesis. Photosynthesis is, on the face of it, very simple. Sunlight energy to food. But when you get into the details of it, it's quite complicated. So I will try to go slow, and please speak up when things aren't clear. So photosynthesis, all life requires a constant input of energy. Remember the second law, that if you don't have a constant input of energy, chemical reactions go to equilibrium and the organism dies. Source of energy is the sun. Green plants are the main producers, and animals are consumers only. animals eat plants, or they eat each other, and if they eat each other, whatever they're eating first ate plants. Now, I just want to point out that sunlight is not the only source of energy for living things. It's rare that it isn't, but I thought you should be aware there are other ways to sustain life. The alternative to photosynthesis is chemosynthesis. And I have just one slide here on that. If you go down to the yellow print, chemosynthesis is the production of organic compounds by living organisms using energy derived from reactions of inorganic chemicals. So they don't need sunlight to make useful chemical energy. This type of life is usually found deep in the ocean where there is no sunlight, and it occurs in these hydrothermal vents, which are very hot. So carbon is fixed using energy from chemicals such as sulfide. The inorganic molecules are hydrogen sulfide, carbon dioxide, and oxygen. And it turns out that photosynthesis and chemosynthesis have a lot in common. They both need an energy source, they need carbon dioxide, and they need water in order to produce sugars. Now, the difference is photosynthesis produces oxygen, chemosynthesis produces sulfur. So the drawing here has photosynthesis on the left and chemosynthesis on the right. So photosynthesis requires carbon dioxide and water. It makes sugar, glucose, and releases oxygen. Chemosynthesis also requires carbon dioxide and water, but also hydrogen sulfide and oxygen. Oxygen goes in. And what comes out is carbohydrate, usually glucose, and sulfuric acid or metallic, or not metallic, but sulfur, sulfuric acid or sulfur itself comes out of chemosynthesis. Now, I have a couple of pictures here of organisms in thermal vents. They're very strange looking, very strange metabolism. And my whole point here is that you should be aware that these things exist and that sunlight is not necessarily the only source of energy for life. Any question about that? Okay, summary of photosynthesis before we get into the details. Sunlight comes in, it's captured by structures called thylakoids. We'll talk about them later. Sunlight activates electrons such that water is, in order to do that, water is split and out comes oxygen. And then there are two free hydrogen ions. These are transferred through the redox reactions to what's called the Calvin cycle. And we'll go through the Calvin cycle later. But the upshot of it is that carbon dioxide and water go into the chloroplast and out come oxygen and sugar, usually glucose. The reaction on the left is called light dependent. It occurs in the thylakoids, requires sunlight to get started. The Calvin cycle is light independent. It occurs in darkness. So after the sun goes down, the Calvin cycle can keep running. And the Calvin cycle occurs not in the thylakoids, but in the stroma, which is the space between the thylakoid membranes.
And where's that, the thylakoid membrane?
where is the thylakoid membrane?
Yes.
I'll show you a detailed drawing of this later. Thylakoid membranes are really stacks of membranes, which are called grana. And within the membrane itself, within these stacks, is where light is captured. That's where chlorophyll is. Light is captured, water is split to release oxygen and electrons. So that happens in the green part of the cell. The Calvin cycle occurs, it's still in the chloroplast, but it's not in the green part, doesn't require light. And so plants can keep making sugar even after the sun goes down. They don't have any source of energy. Okay, contrasting photosynthesis and respiration. Photosynthesis is really glucose synthesis. It's a metabolic pathway that is primarily anabolic. That is, it requires energy. So to review, carbon dioxide and water make glucose and oxygen. Respiration is the opposite of that. It's glucose, not glucose synthesis, it's glucose oxidation. And it's primarily catabolic. So it releases energy, starting with glucose and oxygen are used to produce carbon dioxide and water and energy. That's the whole point of respiration, is to derive energy for the cell to do work. Okay, redox reactions. Energy is harvested through electrons that are moved from one molecule to the next. Electrons can carry energy, some of which will be transferred to intermediates as they go down on their so-called downhill journey. The basis for electron transfers is called redox reactions. And that will become clearer in a minute. Now, some substances attract electrons more strongly than others. A substance that loses electrons is said to be oxidized. If it gains electrons, it's said to be reduced. And oxidation and reduction always occur in pairs. Or another way to say that, if one occurs, the other one also occurs. And this you've seen before, but it's a simple illustration comparing oxidation and reduction. So on the left, you have molecule A reduced. It has an extra electron. If it transfers that electron to B, it is now oxidized. A is now oxidized and B is reduced. And that's what you see on the right-hand side. Now, to put this into life terms, we can start with an organic molecule, and let's say that organic molecule is glucose, and glucose is reduced. It has a couple of extra electrons. It can transfer those electrons to what's called an electron carrier. In this case, NAD plus. Now, I'll talk about NAD later. But this is an electron carrier, and it's an essential part of redox. So that after that happens, glucose is now oxidized, and NAD is reduced to NADH plus a proton. So it's a reduced electron carrier. So this would be the simplest explanation for biological oxidation. Okay, there are three possible ways to make ATP from ADP. Substrate level phosphorylation, which, and these terms don't mean anything to you now, but that's what we're going to talk about next. But they are substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. So in the case of substrate level phosphorylation, there's one molecule of substrate gives rise to a dephosphorylated form and a molecule of ATP. In case of oxidative phosphorylation and photophosphorylation, the electron transport chains, these electron carriers, are used in order to make ATP. So first, let's look at substrate level phosphorylation. And as Scott was saying earlier, this occurs in the mitochondrion. It's a direct enzymatic transfer of phosphate from a phosphorylated molecule to ADP to make ATP. The phosphorylated molecule shown here, phosphoenolpyruvate. You can see in red the phosphate group. So we haven't talked about pyruvate. What is that? Well, pyruvate is glucose, which has 6 carbons, that has been split in 2. So that there are now 2 3-carbon molecules, and those 3-carbon molecules are pyruvate. So phosphorylated pyruvate, along with ADP, can undergo the transfer of the phosphate to ATP. So it's a transfer from one pyruvate molecule to another and generation of one molecule of ATP. That is substrate level phosphorylation. Now, the second way to produce ATP is photophosphorylation, which produces glucose in the chloroplast. But it also, in order to produce glucose, it produces ATP. So we start with light energy, which is intercepted by the pigment chlorophyll. Water is split into a half an oxygen molecule plus 2 electrons, which are highly energetic. They're excited. Then they travel down a so-called electron transport chain, and I'll show you in a minute how they're transferred back and forth between electron carriers, and with every transfer, ATP is made. So that the final electron acceptor in photophosphorylation is NADP, and it's converted to NADPH. And the energy from that goes to make glucose. So the point here is that to produce one molecule of glucose, 18 ATP molecules must be produced, and they're produced in this electron transport chain, this downhill journey of electrons, releasing energy at every step. So that's photophosphorylation, produces glucose in the chloroplast. And it does so using this process called chemiosmosis, and I will talk about that in a little bit. This illustrates one step in an electron transport chain, where you start with a molecule A, which has 2 extra electrons. It can transfer them to NAD, leaving just A without electrons, and converting NAD plus to NADH plus energy. Then NADH can get converted back to NAD plus with the formation of another molecule, B, and electrons. So this cycling back and forth with the release of energy each time the cycle turns is a step in the electron transport chain. And that brings us to the third way to make ATP, which is oxidative phosphorylation, where ATP from glucose generates energy, the ATP from glucose, via respiration in the mitochondrion. So now, oxidative phosphorylation is not in the chloroplast, it's in the mitochondrion. Now, electrons from organic compounds like glucose are passed through a series of reactions, which I've just been talking about, called electron transport chain, to inorganic molecules like oxygen. And during this process, there is another process called chemiosmosis, which generates the ATP as energy is released. And you get many ATPs. Remember I said from photophosphorylation, you get 18 ATPs for every electron transport chain that's used. And those are used to make one molecule of glucose. Oxidative phosphorylation in one cycle makes about twice that many ATPs. So let's start here on the right with glycolysis, which results in glucose. And as I said earlier, glucose can be split into two pyruvic acid molecules. And when that happens, two ATPs are generated. And also, two electrons are given to NADH, which then can pass them on. And the pyruvate molecules are converted to acetyl-CoA. And acetyl-CoA is a critical step, a critical compound, because it's the first step in the Krebs cycle. And again, I'll talk about the Krebs cycle later. The Krebs cycle occurs in mitochondria. So we have these electrons. If you come down to the on the lower left side, you get from one molecule of glucose 10 NADH plus 10 electrons. And these electrons then go into the electron transport chain and chemiosmosis. So this is the stepwise process where redox reactions keep occurring back and forth, and with each one, a bit of energy is released. So at this point, the electron transport chain, starting with one molecule of glucose, yields 34 molecules of ATP. Now those 34, along with the 4 others that are produced earlier, gives a total of 38 ATP molecules from one molecule of glucose. So let me try to explain further how this happens. The requirement is a process known as chemiosmosis. And the definition of chemiosmosis is the movement of ions across a semipermeable membrane down their electrochemical gradient. And in phosphorylation, the ion in question is a hydrogen ion. So on one side of the membrane is a high concentration of ions. On the other side is a low concentration. And as these ions move from high to low concentration, ATP is produced. So it goes from potential energy on one side, kinetic energy in the membrane yields ATP. So chemiosmosis is happening as these electrons are moving down the electron transport chain. Are you making the connection between all of these steps? It's quite complicated. Okay, well, we'll go on. So this is a chart comparing oxidative phosphorylation and photophosphorylation. And I find this quite useful in understanding the similarities and the differences between them. The oxidative phosphorylation produces ATP using enzymes and oxygen. It's the last stage of aerobic respiration. Whereas photophosphorylation is the process of ATP production starting with sunlight. Photophosphorylation turns sunlight into chemical energy. The source of energy, molecular in oxidative phosphorylation, molecular oxygen and glucose are the energy sources. Whereas in the case of photophosphorylation, it's sunlight. Oxidative phosphorylation occurs in the mitochondria. That's respiration. Photophosphorylation occurs in the chloroplast. Two distinctly different organelles doing two distinctly different processes. Occurrence, the oxidative phosphorylation occurs during cellular respiration, the breakdown of food to energy. Photophosphorylation occurs during photosynthesis, the buildup of food using energy from the sun. And the final electron acceptor in the case of oxidative phosphorylation is oxygen. Whereas, as I showed you in the diagram, in the case of photophosphorylation, it's NADP, which becomes then NADPH. So this is something, I haven't put this slide set in Canvas yet, but I will immediately after this class, along with the recording of this lecture, so that you can take your time to study these things and think about them. Okay, now we're going to talk about the Krebs cycle. I referred to the Krebs cycle earlier. Here you see it. In oxidative phosphorylation, the Krebs cycle plays a very important role. And now we're going to dig into what the Krebs cycle really is, what it does. So it starts with pyruvate. And remember, pyruvate is simply one molecule of glucose split into two molecules, each with 3 carbons. So the Krebs cycle starts with pyruvate. And pyruvate is converted into acetyl-CoA. I put a circle around that because that's sort of a central compound in much of metabolism. And it's the starting point for the Krebs cycle. So you have the Krebs cycle represented here twice, once on the right and once on the left. On the left, it shows, starting at the top with acetyl-CoA, shows the structures of all the molecules that are involved in a turn of the Krebs cycle. And it shows where ATP is generated. So the function of the Krebs cycle is to oxidize derivatives of glucose, fatty acids, and amino acids to carbon dioxide through a series of enzymatic steps. So the sources for the Krebs cycle can be almost any kind of food: carbohydrate, fatty acid, fat, or protein. And the result of the Krebs cycle turning is that high energy electrons from these oxidized fuels are transported by activated carriers, NADH and FADH, to the electron transport chain. And you remember what the electron transport chain does. It goes in this stepwise downhill direction. And at every step, there's a redox reaction and a release of energy. Now, the Krebs cycle is both anabolic and catabolic. It's catabolic in that it tears organic molecules apart in order to extract energy, but some of the intermediates in the Krebs cycle are used as precursors for other molecules. And so in that sense, the Krebs cycle can be anabolic as well. So that's the Krebs cycle. This is respiration. This occurs in the mitochondrion. Now, just to make the point, these catabolic pathways can start with food, and food can contain proteins, polysaccharides, and lipids. Also can contain nucleic acids, but they're not a source of energy. So these polymers can be converted to monomers. These different monomers, amino acids, monosaccharides, and fatty acids, can all get converted to acetyl-CoA. And acetyl-CoA is the first step in the Krebs cycle, which is also called the citric acid cycle. And as that cycle turns, ATP is generated. So that's how you get energy from the food you eat. The only point of this slide is to put all the three different ways of making ATP in the same slide so that you can compare them directly. Don't need to go through them all again because you already have my commentary on these three ways of making ATP. Now this is another useful chart for comparing the two major cycles that we're talking about. The Calvin cycle, which occurs in photosynthesis, occurs in the chloroplast, and the Krebs cycle, which is respiration, occurs in the mitochondrion. Okay, definitions. Krebs cycle is part of the aerobic respiration process. The Calvin cycle is part of photosynthesis. Krebs in the mitochondria, Calvin is in the chloroplast. Krebs is catabolic, tears things apart. Calvin cycle is anabolic, it builds things up, makes food, makes sugar. The Krebs cycle, ATP and carbon dioxide are produced, the result of respiration, whereas in the Calvin cycle, they're used. ATP and carbon dioxide are used, and the ATP, of course, comes from the light reaction of photosynthesis. The Krebs cycle takes place in all aerobic organisms, whereas the Calvin cycle occurs only in plants. You think about why? Well, it's because it occurs in the chloroplast, and only plants have chloroplasts. Only plants have chlorophyll, the pigment. The Krebs cycle oxidizes glucose. In other words, it takes it apart, releases the energy in its bonds, whereas the Calvin cycle makes glucose. And the Krebs cycle occurs in the presence of oxygen, requires oxygen. Oxygen is not involved in the Calvin cycle. So again, this is something that it will be worth your while to look at this again and think about these differences and what they mean in the overall production of food and the use of food. Now let's come back to the electromagnetic spectrum. How can we capture and make use of energy that's available? This is a review. So we've been over this before. You remember that the electromagnetic spectrum is a series of waves ranging from very long radio waves, which can be a kilometer or more, many kilometers, to very energetic gamma rays. Most of these wavelengths we cannot see, and because the visible light is this white area in the middle, and that can be diffracted from red colors through yellow, green to blue colors. Now plants, as I'll show you in a minute, I'll show you why this is true. Plants can absorb blue color and red color. They cannot absorb green color, green wavelengths. So they reflect it. And because they reflect green, when we look at a plant, that's what we see. We see green. And this is why. Here you can see the structure of chlorophyll A. There are two basic forms of chlorophyll, A and B. And you see the absorption spectra of these two different types of chlorophyll according to varying wavelength from 400 nanometers to 700. So you can see that chlorophyll B absorbs between 400 and 500, and again between 600 and 700, but not between 500 and 600. Similarly with chlorophyll A. And so this is why plants are green, because they cannot absorb light, they cannot extract energy from light at the green wavelengths. So they reflect it instead. Again, just like to point out a cofactor, a magnesium ion, magnesium is a metal, is required for every molecule of chlorophyll. Where in a plant does photosynthesis occur? Well, it can occur in any part of the plant that is green. For most plants, that's mostly in the leaves, but it can also occur in petioles, stems. It does not occur in roots. Roots almost never, if ever, have any chlorophyll. So in a leaf, you can see the structure of a leaf here. It has an epidermis on each side and what are called mesophyll cells in between the two epidermal layers. And there are stomates for breathing. This is how oxygen is released and carbon dioxide is taken in. And if you look at just one of these cells from the mesophyll, you can see that it has a cell wall, has a nucleus, has a huge vacuole, and it has these green objects called chloroplasts. That's where the chlorophyll is. That's where light is captured. The chloroplast structure. This comes back, there was a question earlier about what are thylakoids and what is the stroma. This slide answers both of those questions. So if you look at the chloroplast, this is one chloroplast taken from one cell, the mesophyll cell. And you can see that the chloroplast has a membrane, an outer one and an inner one. And it has what are called grana. Now the grana are stacks of thylakoid membranes. So this is one grana, three thylakoid membranes. And within each thylakoid membrane, there is a compartment. Now between the thylakoids, between these grana, these stacks of thylakoid membranes, is a space called the stroma. Now the grana, since that's where the chlorophyll is, that's where the light reaction occurs. And when sunlight falls on a leaf, it strikes the thylakoids, and that starts the light reaction. The stroma, the space between the thylakoids, between the grana, is where the dark reaction occurs, and that's the Calvin cycle. Calvin cycle occurs in the stroma, and it does not need light, so it can operate as long as it has the energy to do so. Stages in photosynthesis. The Calvin cycle is where so-called carbon fixation occurs. Again, this on the left is this overview diagram where you see in the chloroplast, water and carbon dioxide go in, oxygen and sugar come out. The Calvin cycle is diagrammed off to the right here. The Calvin cycle is a set of chemical reactions, the so-called dark reactions, that convert carbon dioxide to glucose. And it does so by using energy from ATP and NADPH from the light reaction. So the light reaction feeds the dark reaction. And it does so by using a series of redox reactions, the so-called electron transport system. It's a stepwise process. The Calvin cycle is necessary to capture energy. Without it, if it were instead of a stepwise reaction, if it were a one-step reaction from carbon dioxide to sugar, it would be explosive and the energy would be lost to heat rather than converted into useful chemicals.
Sir, did you hear me?
I can hear you.
Okay, I have a question, please. Some leaves just get like green, and some of them like yellow. I don't know. You say just reflect the green lights, whatever. And how come there's some leaves just like yellow or different colors?
Very good question. Why are some plants yellow? Well, they're yellow because in addition to producing chlorophyll, which is green, they produce other pigments like anthocyanins for other purposes. But the yellow simply masks the chlorophyll. So a yellow plant is really a green plant, but you can't see the green because the green is masked by yellow. Now that's true for most yellow plants. Some plants are yellow because they're parasitic. They really don't have any chlorophyll, but they can't live on their own. They have to attach themselves to a green plant for their food source. So if you see, and around San Diego, you will often look out on the hillside and you'll see what look like yellow plants, sort of viney kinds of yellow or orange plants. Those really aren't green plants. Those are parasites growing on green plants. But
is that the dodder that you're talking about?
Yeah, dodder is an example of a parasitic plant. But you make a good point about when you see a yellow plant, if it's growing on its own, If it's not attached to another plant, then it is photosynthetic. It does have chlorophyll. Chlorophyll is just masked by yellow pigment. Sometimes you see orange plants, or you probably all are familiar with red maples. Red maples have red leaves, but that's simply an anthocyanin pigment masking the presence of the chlorophyll.
Okay, got it. Thank you, sir.
Okay, the next few slides I think are pretty much a review. We're almost out of time. Review of what we've gone over so far. So in the light reaction in the thylakoids of the chloroplast, you have a photon of light coming in. It's intercepted by a pigment. In this case, it's called P680. And this is photosystem 2. So the electrons are excited. They go through this electron transport chain. And in the process of doing that, ATP is generated. And then another photon of light is intercepted by a different pigment. And these electrons are excited again, go to a high energy state. And again, high energy compounds are made, NADPH, and you again have redox reactions leading to ATP production. So there are 2 light reactions in photosynthesis. That's the take-home message of this slide, photosystem 1 and photosystem 2. Beyond that, don't worry about the details of these 2 photosystems. And this is, again, more detail. It simply muddles up your mind. You don't need to know this. And so this is a good review slide, the overview of light reactions, where we're taking a bit of thylakoid membrane. You see this little box here? That's a bit of a thylakoid membrane, which is part of a grana, and expands it. And you can see the flow, light, these yellow squiggly arrows are light photons coming in. They excite electrons, and this occurs within the thylakoid membrane itself. This is a bipolar membrane, just like other membranes in the cell. And the electron transport chain occurs within the thylakoid membrane. So that's important to remember. And you have the 2 photosystems, 1 and 2. Again, they occur within the thylakoid membrane. And after photosystem 1, the energy, the electrons, are exported to the Calvin cycle. So all of this depends on light. But after the electrons leave the thylakoid, they stay in the chloroplast, but now they're in the stroma. And that's where the Calvin cycle occurs. So remember to associate the Calvin cycle with dark reactions of photosynthesis. Well, the main thing I want to mention here is this process called chemiosmosis, because that is critical to the generation of ATP during the electron transport chain. So here we're again talking about ATP production. You have these 2 pigments, P680 and P700, photosystem 2 and photosystem 1. Light comes in, excites electrons. but they're very energetic. They go through the electron transport chain downhill, releasing energy. Well, how do they release that energy? Well, they release it by chemiosmosis. Remember, this is a membrane. This is a thylakoid membrane. And so that membrane is right here, and it has a high concentration of electrons on one side of it and a low concentration on the other. And as these electrons pass through the membrane, they release energy. So the electron transport chain going downhill uses chemiosmosis to generate energy. And again, photosystem 1 gets more energy from light. It's more excited electrons, which are transferred out of the thylakoid into the stroma, where the Calvin cycle occurs. So what I want you to remember about this is what chemiosmosis is. Chemiosmosis is the mechanism by which an electron transport chain makes energy. So to restate the definition, chemiosmosis is the movement of ions across a semi-permeable membrane down the electrochemical gradient. So they're moving from high concentration to low. An ion gradient is potential energy that can be used to power chemical reactions when the ions pass through a channel, or in other words, through the membrane. Okay, I hope you make that connection that electron transport chain involves chemiosmosis. An overview of the light reactions. Oxygen is produced because water is split. Oxygen comes off as molecular. So you have H2O. Oxygen comes off as molecular O2. And the two hydrogens are energized to go into the production of energy. Solar energy is converted to chemical energy in the form of ATP and NADPH. And these molecules are used to power the light-independent reactions, which are the Calvin cycle. The light-independent reactions, these occur in the dark. They are the Calvin cycle. Incorporates carbon dioxide into sugars. That is called carbon fixation. So carbon dioxide gas, plants take up carbon dioxide gas. They don't take up enough of it to prevent pollution, but they need it. It's incorporated into an organic molecule, usually a carbohydrate, usually glucose. Glucose is produced, and it can be built up into polysaccharides. For example, plants make starch. Animals might make glycogen. And polysaccharides are a form of storage. They store energy. The Calvin cycle overview. ATP and NADPH from the light reactions power the cycle. Carbon dioxide is incorporated into sugar molecules. As I said on the previous slide, that's called carbon fixation. So what happens is energy goes in, ATP, along with carbon dioxide, and out comes glucose, sugar. Calvin cycle overview. Again, yeah, you don't have to know the structures or what all these molecules are. You just need to know what the Calvin cycle does. and why it does it. And I think this is the last slide. It's one you've seen already several times. It's a summary of photosynthesis, water and carbon dioxide in, oxygen and sugar out. And this all occurs in the chloroplast. In the chloroplast, remember to make a distinction between the light reactions and the dark reactions, and that the dark reaction is basically the Kelvin cycle. That's respiration, that's catabolic. Excuse me, it's anabolic, it builds up sugar. Okay, yes, that is the last slide. Any questions about, yeah, I've used up all the time. Any questions about this slide set? I will put it in Canvas so you have access to it. I'll put the recording after it's processed. I'll put the recording in I'll send you the URL for the recording. And I will try to get a worksheet prepared and send it to you so that you can be looking at it and thinking about it before Monday. And on Monday, we'll talk about the details of deriving energy from food, respiration. Anything else before I end the session?